next we have Bioxytran Inc. They're an OTCQB company trading under the ticker BIXT. The company is a clinical stage biotech with 3 revolutionary platform technologies that were quickly developed using the power of AI. They have a broad spectrum oral antiviral drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that not only neutralizes COVID-19, but also neutralizes influenza and RSV and prevents it from entering cells. They are currently recruiting patients for their dose optimization study, which will give them the ability to proceed with their registrational trial in India after they finish. They have a second platform drug, an oxygenation molecule, being developed to treat stroke, and its development is supported by an FDA-approved device that detects local tissue oxygenation. So due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. And the company has a third platform technology that's capable of treating cancer metastasis. So over the past year, Mike, you have had quite a few updates regarding your licensing and partnership initiatives with big and small pharma. Now I understand today you want to give investors a peek at your virology presentation. So welcome back to the conference, Mike. I'll let you get to it. And after you're done, I think we should talk a little bit more about your pivot to universities.
Okay, well, thank you. And I'm happy to be here again on the Emerging Growth Conference. So we'll start off with Bioxytran. I'm the Chief Communications Officer for Bioxytran. And let's look at our tagline, breaking the one virus, one antiviral paradigm. This is how this presentation that I'm giving is going to be given to the big pharmas of the world. So that's the background that I want to give to you. So here's how we start off. We have a historic discovery in virology, and you'll see there's this idea of one drug and one virus concept, and it dates back into the 1970s, and so basically after 50 years, prolactin M, it's basically debunked that. You see, because prolactin M has broad spectrum antiviral activity. It's in COVID-19, RSV, influenza, and then we also have a patent pending on 60 viruses. So we really thought this through. And if you're kind of curious what this is, it's a historic discovery. And the discovery is pretty much viral, and you read there about the discovery, viral adhesion to cellular membrane. It's the first step in this process of viral infection. And these galectins, and we're going to talk more about that later, they facilitate this process. And you see, what happens is the cellular membrane, it's kind of slippery. It's glycosylated, which means it's covered in sugar. And what else is covered in sugar are the spike proteins. So you have these two slippery molecules trying to connect with each other. Well, how do they do that? The answer is they don't. They need an adhesion molecule, and galectins are basically this viral glue that tethers the virus to the cellular membrane. So that's our big discovery, and we believe it's universal to viruses in general. Now, existing antivirals, they work on this concept of, basically one virus or maybe a couple mutations of virus for one antiviral. And what they do is they disrupt the life cycle of the virus once it's inside the cell. We're different. We stop it from getting inside the cell. And the truth is, they say broad spectrum. There's broad spectrum antivirals out there for like influenza A and B. That's not broad spectrum. We're talking about broad spectrum that goes across multiple viruses, multiple viral families, and no broad spectrums like that exist today. And we believe prolactin M is going to be one of the first. And why this is a big deal? It's disrupting a $55 billion market, and that is why these drug pharmas that we're going to be presenting to, that's why they're going to pay attention. So we start there. And then we give them an overview of our platform technology. So we explain to them it's carbohydrate chemistry. COVID-19 was our first case study. And then we have an active FDA IND right now. And you'll see the graphic right there in the center. These are a whole bunch of families of viruses. And what we did is we kind of circled them, and you can kind of see, Herpesviridae, that happens to be shingles. And then you got influenza and bird flu, that's Oxymoxiviridae. And then, you have these other two. So there's 4 different families of viruses, and galectins are implicated in them. And you'll see that there's a galectin signature to each one of these viruses. You see how there's up arrows and down arrows. And, there's also a link there to the Science Journal article. So when we're presenting to Big Pharma, everything is backed up. We think that we're just touching the iceberg here, and we think that we should do testing in all of these families of viruses eventually. We start with in vitro testing, then do animal testing. So now it's important that we get into the mechanism of action. Prolactin M neutralizes the spike protein. And, this just demonstrates how it works. So you have the naked, this is a COVID spike, the naked COVID spike, but that's not how it exists in nature. How it exists in nature is covered in sugar. And the reason it's covered in sugar is so that it can hide from the immune system. The immune system sees that spike protein, the protein that's going to go after, it wants to, it sends an immune response after it. So when it's shielded like this, it's protected from immune attack. And that's how the virus kind of tricks your immune system. But you can see there's a circled area there in red. That's the galectin fold. You see how it's uncovered in light blue? And then you'll see the tip of the spike protein. So what you're seeing there is those are the vulnerabilities in the spike protein, or places where the immune system can attack or attach to. And our molecule, we believe, attaches in the galectin fold, and that's the process of neutralization. This next slide here is our antiviral drug discovery platform. And we're basically using nuclear magnetic resonance imaging spectroscopy to check for the binding affinity of our molecule to the different spike proteins. So I'm not going to, go over this too much, but that's the essence of our technology and how we can develop new drugs. This is the mechanism of action. And again, this is all peer-reviewed stuff that the pharmas are going to look at and really, get into. What you'll see there, though, in the upper right is you'll kind of see how the galectins, they facilitate this tethering or viral attachment to the cell. And you can kind of see the first shows the green if it's attached. And the second shows, well, what if we use an inhibitor? Then it can't attach. It's a really simple concept. And I wanted to highlight one thing that we'll probably talk about later is hemagglutinin, the second bullet point. Hemagglutinin spikes, they're involved in influenza and bird flu. Okay, so just keep that in the back of your mind. Now, pharmas, they're not easily convinced. So you got to basically hit them over the head. And there's this one article here that really smacks them. The pivotal role of galectin-3 in viral infection, a multifaceted player in whole that's a word salad sandwich. The bottom line is right there. Galectins are a universal druggable target for viruses. Universal druggable target for viruses, and they're involved in the complete life cycle of the virus. So no matter what stage of the infection, of a viral infection you're in, they're involved. So you know how some antivirals, you got to take them like Tamiflu, you got to take it within the first 48 hours, or otherwise the benefits very limited. Based on this theory, that's different. So I just wanted to, this is something that we would highlight because there's more utility to this drug over the current antivirals. So we think this is a paradigm shift. Now, these are slides that big pharmas are going to want to look at. They're going to want to look at your in vitro data. We're just going to skip through this kind of fast because I don't want to get too science. I don't want to get too deep into the science with you guys, but you can kind of see prolactin M in vitro and RSV and the other one. And then we get to the clinical trial results summary. This is where we're going to focus a lot of our time and attention with the pharmas. Number one, elimination of viral load and symptoms within 5 days. We reduce infectivity, so that means we're not spreading it from one another to each other. And we're going to quiet this. We're going to quiet the cytokine storm. That's the theory behind it. And we're going to have a robust antibody response, meaning that just because we clear the virus so incredibly quickly, that doesn't mean that you're going to be subject to follow-on infection because you didn't build up an antibody response. You're actually building up a more robust response. Now we get into the actual clinical trial results. Now on the left, you see some lines there. The control group is red, and the green group is the treated group. And you'll see a horizontal red line. So you want to get above that horizontal red line as quickly as possible in this particular situation where we're testing CT values. And what you'll see here is on day 3, 88% of the people were PCR negative, and on day 7, 100% were. And you can kind of see how there's a grouping between the two things. And then down below is kind of a summary of where the control was in relationship to the treated arm, which is in blue. And again, peer-reviewed journal articles, so we stress that. And then this slide is going to be, this slide's a moneymaker because, Paxlovid is the standard of care. Now, obviously, should we send this to Pfizer? We're not going to put this slide in there and just kind of beat them over the head. But for the other pharmas that we're talking to, we can show this. And it basically shows a comparison where you end up at the 30% line. All right. Next slide. Pharmas are highly sensitive when it comes to improvement in symptoms. So they want to see symptom improvement, some sort of clinical improvement score. And you'll see that we do that. And again, it's in the literature. And then there's, what about viral rebounds? Well, Paxlovid was common after day 10, and there were none for us. And you can see in the second half of the slide. And then this slide is going to be interesting. This gives investors a little peek into what we're thinking and what sort of money is going to need to come out there to move forward with our trials. And you can just take a peek at it. The one thing I'd highlight is probably, you have the IND. The number 2 is my favorite in the sense that I think when we build out the in vitro studies, people are really going to understand the magnitude of what we have on our hands. We have intellectual property, and then we have a licensable drug, basically. It's oral, chewable tablet, easy to take. We don't think there's any limitations on who can take it. We completed a phase 2. We are ready to start. We have an IND. We can upgrade once we have some PK studies. And there's no safety signals to date. And another one that the big pharmas are going to look at It's like, what the heck is DDI? No DDI required. That is drug-to-drug interaction. So basically, we don't have to go testing. Well, what if you take this drug and that drug? You know, is there an effect? Because this is an untreated population. No one's going after this population except us. And then it's inexpensive manufacturing, easy to source the active ingredient. I think that is pretty much the end of my presentation to Big Pharma. Let me see if there's anything else here. Yep, that's the conclusion. So it's kind of back to you, Anna.
Perfect. Well, as always, Mike, it was an informative presentation, and I hope for your sake that this eventually translated into some sort of licensing deal with the pharmas.
Mm-hmm.
So I told you I wanted to circle back on your latest press release with the University of Georgia. So understanding it a little bit more, they are on the top university when it comes to dealing with poultry health, and some consider Georgia the poultry capital of the United States. Yep. We know this is a big issue here in the US. Most of our chicken and eggs, they come from farms in Georgia. We love our chicken and our eggs in the US. So talk a little bit about this collaboration. How did it come into play, and what are your goals from it?
Well, you know, we actually had a shareholder who brought this to attention and made an introduction. I believe we have the best technology, bar none. So it kind of makes sense that we would team up with the top university, like the University of Georgia, which is actually world renowned for its study in bird health. Now, if you go back and read my LinkedIn article on bird flu back in April of 2023, you're going to see we were very early to not only recognize that bird flu was a big threat, but we also proposed a common sense way to treat it. You see, the culling of chickens to control the spread of disease, this just wasn't sustainable, which is why the government is looking for a solution. And I'm pretty sure that we have it. Now, they're offering $100 million in grant money to develop solutions, and we're excited about that.
So I heard about that, and apparently the USDA is going to support bird flu projects so they can see if there's a way to treat the birds once they're infected with bird flu instead of culling them all. Millions. It's devastating. So can you tell us more about this program and how it fits in with your overall strategy?
Okay. The APHIS program that you're talking about plans on funding up to $100 million worth of research in order to develop a vaccine or a treatment. We fall on the treatment side of things. So they're looking for better ways to diagnose the chickens and also improve biosecurity. Now, our focus is on the development of a water-soluble galectin antagonist capable of neutralizing the virus in the birds. Now, we think the grants are a great way of bringing awareness of what we do. And don't forget, we have the world's first broad-spectrum antiviral. You know, it's not approved yet, but it has that activity. And I've been trying to preach this concept to investors for a couple of years. And finally, you'll see the stock woke up. But the truth is, that's not saying much because it's still stuck at like a $17 million market cap. Even at the current stock price, we are so massively undervalued, and I hope investors understand that. See, a lot of people believe that COVID's over and kind of put us in this typecast as like a COVID stock. So it took something like a massive rise in egg prices to actually get investors to wake up and see that there's another use case for our broad spectrum antiviral.
Well, and we can all feel it. Egg prices have certainly skyrocketed. We see the shortages everywhere. It hurts our pocketbooks. I saw that you tweeted about that and showed how the price of eggs are basically hitting new highs. And it was actually highly correlated with Bioxytran's stock chart. So what do you make of this anomaly?
Yeah, you know, I saw the chart of egg prices and almost like did a double take. I'm like, I've seen this chart before. And then I looked up our stock chart and I was like, man, this thing is spot on. So This basically means if eggs continue to rise, there's going to be a greater demand for our disruptive technology. That's kind of my takeaway. And our stock should follow in suit, right? Now, it's kind of refreshing to see investors realizing that Bioxytran is basically this blue chip pure play in bird flu. And at the last Emerging Growth Conference, I kind of went over our competition in the human antiviral market, but I really didn't touch on the opportunity in this livestock market. And, there's only one player out there, Zoetis, and they have a potential vaccine for chickens in the queue. But the current administration is just downright nasty toward vaccine makers, and that's really great for us. If they mandate vaccines in chickens, it's basically going to destroy the export market for the chickens, because the other countries that buy chickens, they simply won't buy the vaccinated ones. And we think this is a huge opening for us. If we can show some efficacy, and we will shortly, then I think we'll become the favorite in this billion-dollar livestock business to prevent the spread of bird flu.
Well, you seem really confident that you have a solution in bird flu, and I don't think you've done any testing. So where does this confidence come from, Mike?
Anna, here's the thing. We're talking to the top universities in the world about this problem. And when they see our in vitro data in influenza, they see how robust our phase 2 data is. They get the idea that our galectin antagonists are in fact broad spectrum and target this viral entry that we talked about in the beginning. Now, all the scientists we're talking about, they get it, and they get it fast. Unfortunately, the only people not getting it are investors and government researchers that are basically looking to evaluate our work and how many boxes you check to qualify for their funding program. It's kind of this tale of two worlds. We have a massive buy-in from the world's top university in poultry, but investors are treating us like we're almost a startup. Just look at our stock price. Now, there's a massive disconnect between researchers and investors. Now, one of the virologists that we talked to shared the mechanism of action of some of the solutions that she was researching. And you know what I learned? Almost all the ideas revolved around the concept of modulating the immune system to squeeze out a better immune response from the chickens to the virus. I mean, we tried that approach in COVID, and not one draw saw the light of day. Now, add to this challenge of treating domesticated animals, because why? It needs to be easy to deliver and cost effective. So that's another constraint that you're putting on a solution. Now, compare these other alternatives. We have basically the best of breed solution, and that is how we landed a top researcher. And I'm not sure investors have completely connected those dots yet.
You just mentioned top universities. So does that mean you have other collaborations in the works?
Yeah, we're always looking to collaborate. And I think a number of universities would be well served if they were considering using our water-soluble galectin antagonists in their research. I'm very excited because I really think the pendulum has swung back in our favor. The scientists get what we're doing, and it's just a matter of time before that trickles down to investors. If you're an investor and think that we only have one collaboration in play, that means you haven't been following the breadcrumbs I've been putting out there on Twitter, or you haven't been looking at all the people I've been networking with on LinkedIn either.
So help me understand what's going on with the bird flu strategy. You took your broad spectrum antiviral that works on COVID, RSV, and influenza, and then adapted it for animal use. So you're pretty sure it's going to work in animals. But I think the question investors want to know is whether it will work in humans if the virus makes the jump.
You know, I'm really confident based on theory that the existing galectin antagonists are capable of neutralizing bird flu in humans. The thing is, that isn't a really big market. There's right now only 70 cases of bird flu in the United States. So it's not realistic to do clinical trials at this time. Now, if it does jump to humans, we are going to be ready to pounce since the safety data is out of the way, and it basically enables us to find a quicker pathway to market.
Well, I look at the price of eggs. It's obvious to me that we need a better solution than just killing all the birds. And RFK has criticized regulatory bodies like the FDA and USDA that have overreached and prioritized corporate interests over public health. It has a detrimental impact on the pace of innovation. So, how do you feel about that? Are you concerned about regulatory hurdles? How do you plan to navigate the challenges to ensure your product can achieve timely access to poultry farmers?
You know, the administration is clearly looking for a solution that doesn't involve a vaccine. We have a water-soluble, broad-spectrum antiviral that we think will work in chickens. What we need to do is prove that we have a solution and then optimize it for the chicken farmers. Once we have proof that we can prevent infections, the chicken farmers will line up to buy the product. I'm confident of that. The market will ultimately dictate if they want a feed supplement or a veterinary drug. So we think we're highly insulated from any regulatory issues. If we run into any resistance to one path, we just go the other route.
So how do you plan on making money for shareholders?
You know, that's simple. Licensing deals. We've always been about developing the innovation, and then we license it out.
And how long do you think it'll take to develop your product for bird flu?
You know, the manufacturing facility is already in place. So we just need the proof. I'd say 6 months after funding, we'll have the proof we need, and then we can either market or license it.
So a question from a viewer, could the galectin antagonist be added to raw milk to solve RFK's raw milk with bird flu problem?
No, it doesn't. It doesn't work like that. I mean, I gotta think about that a little bit, but no, I don't think it works that way because milk could have a neutralization property to it.
Another question, is it possible to donate a galectin antagonist to a hospital such as Memorial Sloan Kettering, maybe to run a 10-patient trial, see what happens? Have any mouse trials been done in this manner?
You know, I think we're open to all those sorts of collaborations. So great question there. Yes.
And does prolactin M potentially work on avian flu and EEE? I'm not sure what that is.
I got to punt on that one because I don't know what EEE is.
Okay. And Heather's asking, when could we see prolactin M in the hands of the general public?
You know, again, that's a really good question. It really depends on our access to funding, and and obviously patient recruitment, and the FDA. I think I think what JFK is doing is trying to create greater access for us. So we see that as a positive movement.
Perfect. Well, Mike, do you have any closing remarks for our viewers today?
Guys, I don't know how to pound the table. The valuation, it's incredibly low. We're talking to the right people. You need to look at that valuation and say, does this make sense? And remember the 3 M's of biotech. I've said this before: money, magic, and management. And I'm telling you, we're going to have all 3.
There we go. Great presentation and back and forth as always, Mike. Nice speaking with you, and I look forward to seeing you again real soon.